Hey, Dana, I have a class that starts in 5 minutes. This is gonna be really fast. Okay, so background information, useful information you learned in 7th and 9th grade. An organism has two identical types that is called homozygous. You can be homozygous dominant, you can be homozygous recessive. That's a capital T if you're homozygous dominant. That's a lowercase t if you are homozygous recessive. I'm only talking about Mendelian genetics right now. So this is complete dominance. This is an expression of normal dominance. This is none of the exceptions. An organism that can have two different genes, we call that heterozygous. If a lot of times we're talking about it in regards to disorders, we will also call this a carrier, meaning that they carry the recessive trait. That is not what they are going to express, again, because this is normal Mendelian genetics. Unlike homozygotes, heterozygotes are not true breeding. So another vocabulary word that's going to be important for you to know is this true breeding. A true breeding means you are homozygous. You can be true breeding for a dominant trait. You can be true breeding for a recessive trait. You cannot be true breeding if you get one of each. That's a hybrid. So we've got two big ideas here. We have a genotype. That's the genetic makeup. Those are those two letters, those two alleles that you inherit, one from each parent. We learned that about in the meiosis video. An organism's phenotype, however, is the expression of that. So that's the actual proteins that are made, and that's what it's going to look like, the physical appearance. So in this particular example, we have a phenotype of purple or white. We have genotype options of homozygous dominant, heterozygous, both of which will get you a purple flower, and then homozygous recessive, which will get you a white flower. The reason we like to talk about flowers so much is because this is similar to what Mendel did in his experiment. So another kind of just like embed some more vocabulary in you. We had Mendel do these all based off of math. So he literally just did crosses over and over and over again and looked at the probability that something was occurring. And so what he started with is a P generation. This is the parent generation. And so the parent generation that he always started with was a true breeding purple plant. He figured out it was true breeding because of how many times he just replicated this is purple, 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 and purples only ever gave him more purples. Same thing with whites. He was like white with white, white with white, white with white, until he was only getting whites. And so when he crosses those, what he found out that all the plants were purple. Like you could never cross a true breeding purple with a true breeding white and get a white plant. It was very strange. And so we call that the F1 generation or the future one generation. All of them are typically heterozygous because from the P generation you go and cross two true breeding, you're only going to get heterozygous ones. And then in his F2 generation, meaning he crossed two members of the F1 generation, so two heterozygotes, his ratio ended up being about a 3 to a 1. So a couple things to kind of take away from this. First thing is there are these things that are genotypic ratios, and you write them three numbers. The first number is homozygous dominant, your second number is heterozygous, and your third number is homozygous recessive. So that's your genotypic ratio. Your phenotypic ratio is just two numbers. It's just everything that looks dominant and everything that looks recessive. So for instance, in this particular example, this genotypic ratio was a 0, 1, 0 because every single plant looked or was heterozygous. The phenotypic ratio was a 1 to 0. Everybody looked dominant. And then in this one, we had when you cross here, your genotypic ratio would be 1 to 1, because one of these is actually homozygous dominant, two are heterozygous, and one is homozygous recessive. And so that's your genotypic ratio. But then your phenotypic ratio is going to be a 3 to 1 ratio. So Mendel, doing these experiments, he came up with these 5 big ideas. The first idea that he had was that there are alternative versions of genes. He saw purple, he saw white. He was like, hey, that's the same gene. but it's two different expressions. And so he kind of coined the phrase that they were alternative forms of a gene, which we now call alleles. The second thing was that for each character, an organism inherits one from each parent. So he knows that there are two alleles. He knows you get one from each parent. Now, here are the three laws that he kind of had, and this is probably the extent of your knowledge from ninth grade. If two alleles differ, the dominant one will mask the other. That's called the law of dominance. And so that's why we call it normal Mendelian complete dominance inheritance, is because if we see, a ratio of one masking another, that there's only two options there. That's the law of dominance. Second one is he had the law of segregation. This is basically saying that when gametes are made, there's the bell. I told you it would happen. When gametes are made, they are going to have one from mom and one from dad. And then last but not least, the principle of independent assortment. This is largely in part to crossing over during meiosis. It is not necessarily guaranteed that you're going to inherit all of mom's traits or all of dad's traits. It is a possibility. However, you're going to get a lot of chromosomes doing this. So just because you got mom's blonde hair does not mean you're going to get mom's eyes. And so that basically, because the chromosomes are all willy-nilly when they separate, and because of crossing over, they will assort independently of each other before they separate to do gamete formation. And so that's his 5 big ideas. Mendel did this again by just lots and lots of cross-pollinating. He took true breeding, he crossed those. He did a lot of heterozygous crosses, and we're going to make sure we understand this vocabulary of P generation, which is 2 true breeding, F1 generation, which is cross 2 hybrids, and then the F2 generation was then what he was able to see. Okay, so Mendel Genetics, let's go over the first one so we can actually see it. Alternative versions of genes, you can be dominant, you can be recessive, you can inherit homozygous dominant, you can inherit homozygous recessive, you can be heterozygous, where you get one of each. Second thing Mendel discovered, you get one from each parent. So here's a dad's genotype, here's a mom's genotype for chromosome 23, the sex-linked chromosomes, and you're going to get one from dad and one from mom. And you can be a son if you inherit Y from dad. Or you can get one from dad and one from mom. And you can be a daughter if you inherit the X from dad. So Mendel also discovered the law of dominance, okay, which means the heterozygous individual will resemble a dominant phenotype. So in this particular example, homozygous dominant is red, homozygous recessive is yellow, but the heterozygous individual is also red. This is going to mask, cover up the appearance of the recessive trait, okay? Number 4, law of segregation. So when we look at these alleles in a parent, we go from diploid, where you have one of each, so that a pair, to that you can only inherit one of them. So the gametes only have one. And so if we look at pure breeding, you can only get a T that is dominant from one parent and a T that is recessive from the other. In all of the future generations, you can get this or that, which is how we get these offspring in that particular order. So this is because of meiosis that these alleles separate. Yeah. All right. Next one here, we have another bell. The Mendel discovered also the principle of independent assortment. So really want to zone into this picture for you. Without seeing crossing over, which is the thing in meiosis that helps add genetic diversity, you can see we have two parent chromosomes that are the same. This is dad's, this is mom's, okay? So what I can give to my offspring is I can only give 1 and 1. So in this particular version, they lined up, all dads are over here, all moms are over here, and they're going to split. Okay. And so when we do that, the haploid cells are just dads, just moms, just moms. Versus we can actually independently assort these where dad's here and here. And what happens is when they split, now you have a mix of the two. And so principle of independent assortment is basically there's any hundreds of combinations of those 23 pairs passing on mom or dad in either one. So biggest ones here, we've got law of segregation, law of independent assortment. We went over that and where those things occur. Segregation and independent assortment can be applied to genes that are on different chromosomes. This is because if you have a gene on one chromosome and a gene on another chromosome, then you can split them, and it's just like splitting chromosomes. But also, you can do this to genes that are on the same chromosome if you have crossing over. So we don't apply independent assortment to crossing over because there's a lot of different genes and they all independently assort from one another on different chromosomes. But in crossing over is another way that we can continuously incorporate more genetic diversity by mixing traits within a single chromosome. So, like we said, probability 1 here, all of this is going to get from one parent, all of this is going to get from the other. So this offspring, okay, will get only grandma's stuff. I know that's wild because this is parent, this is child, but this is grandparent and grandmother. So that's where they got their original info from, okay? You can even look at this in multiple generations. Versus probability 2, where they split up and independently assort from one another. Okay. So another and final way that we get genetic variation, and just to kind of like explain this a little bit better, is hi Haley. Haley apologizes for interrupting. Origins of genetic variation among offsprings. These are 3 things that contribute to genetic variation. The more heterozygous we are, the more genetically variable we are as a population. And just this is so you can see the big picture, is the more genetic variability we have, the more adaptable we are as a population. So the 3 mechanisms that can contribute to genetic variation, which again, this is going to be something I harp on more when we get into evolution, is independent assortment of chromosomes, crossing over, which adds to genetic variation, and then obviously random fertilization. The fact that we're not saying this male and this female have to mate. It's any combination of male and female that increases your genetic variation. Crossing over increases your genetic variation. Independent assortment increases your genetic variation. And all of those things can help lead to evolution because we have a lot of different traits that could be beneficial. And if you're a beneficial trait that helps you live longer, reproduce, then the overall population is going to start to evolve that direction. Okay, so to show you how crossing over contributes to increasing genetic variation, here we have in prophase 1, once again, the 2 homologous pairs touch each other at the chiasma, sometimes called a tetrad. The 4 together is called a tetrad. And so what happens is now we have a just dad gene that you can inherit, a mom and dad gene, a mom and dad gene that's the opposite of this one, and then just a mom. Okay, so as you see in your gametes, each of them is genetically unique. And that crossing over can occur anywhere on that chromosome. And that's happening in every single pair. All 23 pairs are getting crossed over and exchanging genetic information. Random fertilization we talked about, the mother and the father, they have their own genetic code, and it can contribute to what they pass on to their offspring. And then last but not least, Oh, actually, I think we're good. Okay, so that's your genetic code. All right, peace out. That was a great